Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Sebastian Schuck from Wacker Biotech.
Yeah, thanks Leah for the introduction, and welcome everybody to our today's webinar, which is on protein refolding by FOLTEC. And today you will enjoy two speakers of Wacker Biotech. First of all, we'd like to introduce the speakers. On the left side, you see our expert Andreas Anton. He is the head of our process development department at Wacker Biotech. He has very extensive experience in manufacturing of biopharmaceuticals in microbial expression system, and he has a strong expertise in protein refolding. My name is Sebastian, and I'm working in the business development department of Wacker Biotech. And first of all, we'd like to introduce Wacker Biotech to you. We just have one slide which shows you Wacker Biotech in a nutshell. We consider ourselves the microbial CMO because, in contrast to many other CMOs, we are dedicated to microbial expression systems and more specifically E. coli. So we are part of the Wacker Chemie corporate group, which gives us good financial and long-term stability. We operate two GMP manufacturing lines, both located in Germany, with 300 and 1,500 liter total fermentation volume, and both are EMA and FDA inspected. In contrast to other CMOs, as I said, we focus on E. coli, and we offer cost-saving E. coli technologies for the benefit of our clients, and they help to reduce and bring down the cost of goods. So on the next slide, a very prominent example of our E. coli technologies is called FOLTEC. You will see it here on the right. FOLTEC is an abbreviation for E. coli secretion technology. This technology is out since almost 10 years now, and it's based on soluble expression of a target protein and secretion into the culture board, which facilitates downstream. And now our new technology, which we termed FOLTEC for E. coli folding technology, can be seen on the left. It is based on refolding of inclusion bodies because many target proteins can be manufactured in a soluble way, but it does not work for each and every target molecule. So now we offer in synergy to our soluble expression platform FOLTEC to offer a complete package to our clients. And what does that mean in reality, and what is FOLTEC really about? FOLTEC is basically based on three pillars. On the one hand, these are proprietary E. coli strains. They are based on a K12 and EcoB background. And in contrast to other expression systems, they can do without antibiotics. So you don't need any antibiotics in the production process, which is in line with regulatory requirements and also safety concerns. And you do not compromise on productivity. So they are still characterized by a very high level of inclusion bodies, have a very high gram per liter yield, and also the quality of the inclusion bodies is superior. You have less host cell impurities, and that is a really important aspect of our new technology. The second pillar is a phage-free plasmid system. In contrast to other common systems, which are T7 systems, for instance, they require phage components to shuttle the genetic information into your host system, and this arises safety concerns. This is not just about reactivation of the phage, which is always an ongoing discussion, but also a matter of false positive analytics. And for a CMO, phages are nothing what you want in your plant. So we have developed a system which can do without any phage components and also without antibiotics. And last but not least, it's a rational design of refolding with extensive experience in protein refolding, and developed a toolbox how to identify the ideal and optimum refolding conditions. And Andreas is now giving you some slides and some data on recent case studies and how FOLTEC looks like in daily doing.
Okay, hello everybody. Thanks for the introduction. Yeah, I would like to give you a brief overview about protein refolding and our new FOLTEC technology. Here on this slide you see one of the first aspects we try to show you. It is the new plasmid maintain system without antibiotics, and you see here on this page part BL21D3, which is a very common E. coli strain for the production of proteins, but as already mentioned, it contains a prophage, and if you look on the genetic stability, it decreases under inducing conditions very fast after already 3 hours, from 86% down to 5% if you go to the 24-hour time point. In comparison to that, we have a very high stability of the plasmid during the fermentation, starting with 100% after 3 hours, and also after 1 day of induction, we still obtain more than 99% of plasmid retaining cells in the culture. If you look on this part of the slide, you see several FOLTEC strains. Here again is BL21 as a comparison. You see here the expression level, and with all FOLTEC strains you get pretty much the same expression level as you know it already from the very common T7 system BL21, but as already mentioned, without the phage and with a high genetic stability in the background. And we also have EcoB strains. BL21 is an EcoB strain. There are several E. coli lines used in the lab because they are derived from several patients in the 20s of the last century. And BL21 is EcoB, but we also have K12 strains in our toolbox, which are used for screening to obtain the best expression level for our products. Yeah, this is an example how the strains work in fermentation. You see here an OD in a 150-liter fermenter in our process development department. We see a very similar growth behavior of BL21 in comparison to one of our FOLTEC strains. Very similar expression level of the inclusion body material. And if you look on the timeline, you see it takes less than 15 hours to come to a pretty high OD and also to several grams per liter titer. So you don't have any compromise in terms of productivity with our FOLTEC strains in comparison to what you already know from BL21. That was the first. So now a little bit about the background of protein refolding. I'm not pretty sure how familiar you are with this approach, but your starting material are always so-called inclusion bodies. These inclusion bodies are formed in the cytosol of E. coli, driven by the reducing nature of the cytosol. So this means reducing nature means disulfide bridges, which are usually used for stabilization of structures in eukaryotic proteins, cannot be formed by E. coli itself. This means the protein cannot form its natural three-dimensional structure, and usually these proteins are prone to aggregate and form these particles inside of the cytosol. What we are doing is we solubilize these aggregates by chaotropes, which means using guanidinium, urea, sodium hydroxide, or detergents to resolve these peptides and bring it to its native. And this means there are several approaches available. What we very often do is fast dilution as a scalable technique for refolding of proteins. And there are two competing reactions always in a folding reaction: folding itself and aggregation. And aggregation as a reaction of higher order is always promoted by high protein concentrations, and folding is promoted at low protein concentrations. This is known. And what we're doing, we try to find conditions where the correctly folded protein is promoted, and we're looking for conditions which promote the folding of correctly folded material. This is how it looks like in reality. This is one of our folding vessels. It's a stainless steel vessel with a specific steel quality because you handle a high concentration of chaotropes, high concentrations of additives in the refolding buffers. And one of the part is to get these processes scalable. And I come to this a little bit later, but if you look on these vessels, this is a 2,000 liter vessel. And if you think about high and low protein concentration, then of course one of the goals is to reduce the folding volume, because if you think about primary titles of 10 grams per liter and think about folding concentrations of 0.1 gram per liter, there's a factor of 100 between folding and fermentation. And if you have, for instance, a 1,000 liter fermenter and you have a factor of 100, you can imagine how huge a vessel for folding should be. And the goal is always to reduce these volumes to an appropriate level, but to keep high expression, higher refolding yields during a refolding step, also in manufacturing. This just should give you a brief overview what kind of parameters are influencing the refolding. And first of all, the expression strain itself is one of the aspects, the expression level, because high expression level means high inclusion body quality. Yeah, all the things are incorporated into inclusion bodies, and what we are trying to get is high expression titles, because the quality of inclusion body material, also the particle size increases with that, and we try to find a way to get high titers. The inclusion body quality itself is also influenced by the washing method, the buffers and the additives you use for washing. And finally, you go to solubilization and folding conditions, and all these parameters you see here are influencing these. Finally, just deal with the volume aspect, because you have to get it into the vessel itself, and to keep approximate or to keep high refolding yields. And this basically influences your process design. An example, when we use this FOLTEC technology, is a regular thrombin. Thrombin is one of the final enzymes in your blood clotting cascade. It converts the fibrinogen to fibrin, as used or produced in your liver as a cymogen. It's prothrombin in your body. It's a pretty large propeptide or protein, and it is activated during if you have a broken blood vessel, and then it's activated. Clinical use is surgery to control this kind of bleeding, and it's a 37 kilodalton heterodimer, and this heterodimer is a chain A and a chain B, and the chain A and B are connected by a disulfide bridge, and within chain B there are 3 additional disulfide bridges. And in E. coli you can use a small precursor as a starting material. It's so-called prethrombin, which is basically chain A and chain B, but these 2 chains are still connected at the backbone of the peptide. The backbone connection is cut during the activation process. We started with very low folding yields of 4 to 7%, and we tried to find a way to improve the refolding of these kind of material, because with 4 to 7% it is usually not suitable to produce proteins from a manufacturing aspect. What we're doing is we subdivide our folding screening in different rounds, and this is just an example. First round is screening for additives. It's a buffer screening, and what we're usually doing is combining several additives with overlapping activities. If you think about additives like arginine, glycerol, guanidinium hydrochloride, urea, they have all different activities in terms of protein stabilization, and we try to find any overlapping activity. And we combine this with a very low protein concentrations and an average protein concentration, so 100 mg per mil and 500 mg per mil. And this gave us the first insight how easy the protein can be folded and what is the protein-related issues with these approach. What we're usually doing at the beginning of a folding screen is we establish an analytical tool which gives us the opportunity to discriminate between folded and unfolded protein. In the case of thrombin, we used an HPLC method, or pHPLC, which basically discriminates between oxidized and reduced protein forms, and gives us the opportunity to orthogonal check these HPLC with a enzymatic assay, because thrombin has a protease activity, and so we followed this folding screening. At the end of the day, after several screening rounds, we came up with an increase of from 4 to 20% of refolded protein. And at the same time, we could reduce the folding volume for production of 1 gram of protein from 30 liters down to 7 liters with our new approach. You see, overall, we had a higher productivity for this project. From our perspective, temperature was critical, as well as protein concentration, and you could identify a very specific new refolding additive, and we obtained higher protein concentration by using so-called pulse refolding, because if you think about refolding and scalability, pulse refolding gives you the opportunity to introduce more material into one folding batch without increasing the volume. And this is already the end of my talk, and I would like to thank you for your attention, and I'm happy to take your questions.
So the first question is, how long does such a refolding development typically take?
Usually, folding development takes 10 to 15 weeks. It a little bit depends on the information you already have about your current protein and the availability of reference material.
And what are the common yields of in vitro refolding?
That the common yields for in vitro refolding differs from, of course, from protein to protein. It can start from 20% of refolded material up to 80 to 90%. It really depends on the protein and how much disulfide bridges are in the protein itself, and of course about solubility of the protein.
Okay. And if anyone else has any questions, go ahead and type them. And how do you start a folding development process?
Yeah. First of all, we have a look on the reference material and start with a development of an appropriate analytical method, which we need to discriminate between folded and unfolded protein. Yeah, and this is usually the starting point. We usually challenge also the analytical method with a second analytical method to get an idea that we follow the correct protein variant.
And how do you validate the quality of refolding?
Yeah, that's a good question. That's a really good question. Well, I guess validation means how we are sure that the protein which is folded is correct. I guess this means validation. I hope so. I would say that we always check the variant we are following during folding development. We check, of course, the biological activity, because this is what we are looking for, to get biological active material, and this is one of the aspects we address pretty early on.
And how many grams of protein can you produce? What's the capacity of your reactor?
Oh, I guess the largest capacity we have here at Wacker Biotech is 1,500 liters, and I guess we could produce in the gram to kilogram range. But this really depends on the protein itself.
And have you been able to refold proteins that normally require an end-linked carbohydrate during folding, but not for activity of the properly folded protein?
No, we never done that before, but I guess it should be no big deal to do that. Here, I was asking for how much do you know about the protein itself, and do you know something about the characteristics of the protein? If you all have a clear picture on that, folding of the protein should be no big deal to get to a condition where the protein is folded. It's just always a matter of the procedure applicable for large-scale manufacturing. And this is these are two different questions.
Okay, these are great questions. And how much batch-to-batch variation can you expect during your FOLTEC method?
Batch-to-batch variation using the FOLTEC method. Usually, if you think about manufacturing, and folding is just one part of this manufacturing process, then you have the same variability as you have it for each biological system. I would assume that a complete manufacturing process should contain USP, DSP, and release. And at the end of the day, you have pretty much the same yields over each batch, because if you have the right in-process controls in place to steer your process, and you of course have to keep in mind how much material you get folded, how the material is loaded to the first and second and third column, and then you get the same variation as you have it for each biological system.
Okay. Well, these have been great questions. Sebastian. Oh, sorry. Go ahead.
Yeah, there's no black-white batch-to-batch variability. You have the same variation as you have it, for instance, for show cells or other systems in place.
Okay, and we have one more question. Is it possible, rather than to have a whole protein, to only have a bioactive part of the protein get folded?
Yeah, of course.
If you've tested it.
That is no problem. That's no problem in general. If the active part of the protein folds as a stable structure. Yeah, if you have just a domain which can be folded or should be folded because this domain contains the active part of the protein, then of course you can do that. But at the end of the day, you also have to know something about the of this domain, because you need to know something about stability under certain conditions, buffer conditions, pH, and so on and so forth. It just is not the only way to get a soluble protein derived from occlusion bodies, because in one moment you have to remove the additives which are stabilizing intermediates, and at the same time you remove the additives. Then you have to transform your protein into conditions which the proteins or in which the protein is stable, and this is a tricky part to get this information really early on.
Okay. Well, thank you. These have been great questions. Sebastian, did you have a couple final words?
Yeah, I would like to thank everybody for participating. We're very pleased to launch this new technology for the benefits of our clients. And if you have additional questions or would like to know more about FOLTEC, simply Google FOLTEC. I think the second or third hit is our case study, or drop us an email. It's info.biologics@wacker.com, and the email address can be found on the bottom of each slide. Thank you very much.
Thank you.
Thank you very much, everybody.